Greetings and welcome to the video. Today I'll be presenting the paper Rational Cell Culture Optimization Enhances Experimental Reproducibility in Cancer Cells, written by Muelas, Ortega, Bendtsen and Westerhoff. I hope you enjoy the video. The reproducibility of results is a major part of the backbone of modern science. However, in recent years it has become an issue as the success in reproducing an experiment's results has shown to be declining. This has been reported on in multiple papers and is a major issue for the scientific community. But why is it so hard to reproduce results? In this paper written by Muelas and the team, they investigate whether the currently recognized culture media standards are adequate for consistent results. So how did they investigate this? By performing a series of assays, they investigated between two cancer cell lines the effect of a glutamine inhibitor on proliferation. They used two cell lines, H358, a cancer cell line that is known to be insensitive to the inhibitor GLS1I, shown here in the color blue, and the cancer cell line A549, shown here in green, which is known to be sensitive to GLS1I. GLS1I works to prevent glutamine metabolism and as such would have a negative effect on cell proliferation. The cells were seeded at a density of 8 × 10 to the 5 cells per well in 1 milliliter of culture media for 24 hours, which is when 1 nanomole of GLS1I would then be added, and the effect of the inhibitor would be measured 48 hours later. So what did they find? What they found was that there was no apparent impact on cell numbers for either cell line, and that while glutamine consumption was reduced in the A549 culture as expected, there was a much greater reduction in the consumption of glutamine in the H358 culture. So why did this happen? They measured intracellular and extracellular metabolites of the cells, and they identified that by 1 hour into the assay, almost 20 millimoles of lactate had been produced by H358, suggesting that a majority of the glutamine in the media had already been consumed. This was confirmed by the glutamine content of the media dropping by more than 70% in both cultures 1 hour into the assay, and being almost undetectable by the end of the assay. So how did they optimize this? By reducing the seeding density to 1.5 × 10 to the 5 cells per well and 3 × 10 to the 5 cells per well for A549 and H358 respectively, as well as tripling the culture media, they observed exponential growth in the control samples. They then also ensured that confluence remained below 80% and extended the assay duration from 48 hours to 72 hours. And then they found that through treatment with GLS1I inhibitor, they reduced the A549 cells by up to 20% without affecting the H358 cells, and that culture environmental changes were minimal. So what did they find from this study? They demonstrated that the initial assay on the commonly accepted culture media standards did not produce consistent results for a metabolic study, and that the inconsistencies in culture were the cause of irreproducible results, and that by optimizing the culture for the assay they were able to achieve consistent results. So where do they go from here? From this study, they recommend that experimental design should not rely on historically set conditions for cultures, as the complexity of a living cell may result in metabolic changes between cell lines. And there should be a greater level of experimental detail provided with published results regarding culture conditions.